Hello students, welcome back. Today we are going to discuss some more concepts from evolution. In last part, we started the evidences of organic evolution. Under that, we are completed the morphological evidences. So in that morphological evidences, we discussed about homologous and analogous organs, vestigial organs, atavism, and connecting links. We are completed in the last part. So today we need to go to discuss about some other evidences of organic evolution. So that is evidences from embryology. Embryology is the study of development from egg to hatching or birth is called embryology. The aspects of embryology which supports the doctrine, nothing but principal thoughts of organic evolution, are similar stages of early development in all the mammals. The embryo of all vertebrates are similar in shape and structure in their early stages. All the vertebrates start their life from a single cell called the zygote. All of them during their life history pass through two-layered blastula and three-layered gastrula stage, and then through fish-like stage with gill slits. All the different aspects of embryology strongly supports the fact that the development classes of vertebrates possesses a common ancestry and have differentiated from one another through the process of evolution. K.E. Von Baer. He is the father of developmental biology. He proposed Von Baer's law in 1828. This law stated that during embryonic development, the generalized structures like notochord in chordates appear earlier than the specialized characters like feathers in birds or hair in mammals of the group. So the generalized structures like notochord. So notochord, it has started in chordate, and this notochord is in mammals converts into the spinal cord. So it is a generalized structure. This notochord we can observe in the early chordates, and when it comes to the mammals stage, early chordates are fishes, amphibians, reptiles, aves, and when it comes to the mammals, this notochord is modified as a spinal cord. Von Baer's law consists of the following principles. The more general features of any large group of animals appear in the embryo earlier than the more specified features. The embryos of different species are more alike than the adults and differ progressively from each other during ontogeny. So ontogeny means embryonic development. The embryo of higher species may resemble the embryo of lower species, but never the adult of the lower species. So the embryo of higher species like human is resembled to the embryo of lower species like fish or maybe amphibian or maybe reptile or maybe bird. but the adult of higher species never resemble to the lower species. So here it is, the embryonic developmental stages in chordates, fishes, salamander, that is belongs to amphibian, tortoise, that is a reptile, a chick, that is a bird or aves, calf, and human comes under mammals. So in these chordates, in first row, that is if you are identify the early stages, this stage, this entire stage, it is almost similar early embryonic stages. When it comes to the adult, that means a fetal stage, and at the time of birth, there is a variation in their forms and structures. So in early developmental stages, the embryos are alike. Biogenetic law. This law is also called recapitulation theory. It was proposed by Ernest Haeckel. According to this law, ontogeny recapitulates phylogeny, which means that embryonic development of an individual repeats all its ancestors through which it has evolved. Diagram whatever you observed in previous slide. So in that, chordates are alike. That means human, when it become a adult form, it repeats its ancestors through which it was evolved. Humans are evolved from birds. Birds are evolved from reptiles. Reptiles are evolved from amphibians. Amphibians are evolved from fishes. So all the characters of fishes, amphibians, reptiles, birds are repeats in its embryonic development. So that is a biogenetic law proposed by Ernest Haeckel. The examples for biogenetic law or recapitulation theory. 1. Occurrence of tadpole larva. It is the larva of amphibian in embryonic development of frog. The tadpole has a lateral line organ and internal gills covered with a operculum. These are the characters of fishes. The characters are lateral line organs and internal gills covered with operculum are the characters of fishes, thus indicating that frog has evolved from such ancestors. The presence of that characters, lateral line organs and internal gills covered with operculum, the presence of these characters in the tadpole indicates that the frogs are evolved from fishes. Second example, the presence of teeth in birds embryos, that is a reptilian character. Reptiles having teeth indicates that birds have evolved from reptiles. So nowadays all modern birds are toothless. But presence of teeth in birds embryos, that indicates that they are evolved from reptiles. Third example, presence of gills or mesonephron's kidney in human fetus also indicates ancestral characters. These are present in reptiles. fishes, and also amphibians. Fourth example, presence of bilateral symmetry in the larva of radially symmetrical adult echinoderms supports biogenetic law or organic evolution. So you may studied that in first PUC, that is body symmetry. So asymmetrical, radially symmetrical, bilateral symmetrical. So but here echinodermids. In that phylum, that is example is starfishes. The larva of a starfish is bilateral symmetry, but when it becomes adult, it is radially symmetrical. That supports the biogenetic law or maybe organic evolution. Similarity in early embryonic development. All multicellular animals during early embryonic development pass through common stages like egg, blastula, gastrula, neurula, etc. The common development shows the common ancestors. So having common developmental stages like egg, blastula, gastrula, neurula, having this common development shows that they have common ancestors. Development of organs. In embryonic development of mammals, the heart has two, three, and four chambered condition. So during development, our heart to develop but first two chambered, then it converts three, and finally it become four chambered condition. Indicates the ancestors we are evoluted from other class animals, that is fishes, it has a two chambered heart. Amphibians and reptiles having three chambered heart. Birds and mammals having four chambered heart. So in that reptiles, crocodiles are having a four chambered heart. So all these characters of the heart is repeated in the development of mammals. That indicates that mammals are evoluted from the ancestors. Kidney in fishes and amphibians is mesonephrons, while in reptiles, aves and mammals it is metanephrons. During embryonic development of reptiles, mammals and aves, the kidney formed as mesonephrons like fishes and amphibians, and later it is developed as metanephrons. Fate of three germ layers. The fate of three germ layers like ectoderm, mesoderm and endoderm is the same in all vertebrates. If kidney is mesodermal in fishes and amphibians, it is the same in reptiles, birds and mammals also. If heart of fish is mesodermal, it is the same up to mammals. That means the heart is developed from the mesoderm layer in amphibians as well as in reptiles, birds and mammals. The brain from fishes to mammals is ectodermal. So brain is developed from the ectodermal layer. So this we are discussed in the human reproduction. So of gastrulation. After gastrulation, the embryo differentiates into three layers: ectoderm, mesoderm and endoderm. From these layers, the organ systems will develop. So in that, in fishes, heart is mesodermal. That means developed from the mesoderm layer. In remaining all phylums, classes, not phylums. Phylum is chordate and it is divided into 5 classes: fishes, amphibians, reptiles, aves and mammals. So in fishes, heart is developed from mesodermal layer, and it is same up to mammals. That means in amphibians are also mesodermal, and in reptiles also heart developed from mesoderm layer. Birds as well as in mammals are also it developed from the mesoderm layer. Similarly, brain. So brain is developed from the ectoderm layer from fishes to mammals. Now, embryological evidences in the plant kingdom. The mosses belongs to bryophytes and ferns belongs to pteridophytes, are highly evolved than algae. The early gametophytes of both are represented by the protonema produced by the generation of the spores. These protonema resemble the green algae from which they are thought to have developed. Higher gymnosperms do not need water for fertilization, but primitive gymnosperms like cycads and ginkgo biloba have ciliated sperms and required water for fertilization, just like the ciliated sperms of bryophytes and pteridophytes. In first PU, you may study that bryophytes are considered as amphibians of plant kingdom because they need water for fertilization to transport their sperms or maybe gametes. So that is a character of bryophytes, as well as pteridophytes are also required water for transporting their gametes. So that is the character of bryophytes. Whereas in higher gymnosperms, they do not require water. But in lower gymnosperms or primitive, that is like cycads and ginkgo biloba. These plants are lower gymnosperms. They require the water for transporting their gametes. So that indicates that the gymnosperms are evoluted from their ancestors of bryophytes and pteridophytes. So it is suggested that gymnosperms evolved from pteridophytes like ancestors, and the cycads represent an intermediate link between the lower plants and the angiosperms, suggested a phylogenetic community. So from that gymnosperms and angiosperms are evoluted. So the embryological evidences are the best, but indirect evidences in favor of organic evolution. So embryological evidences are not direct, they are indirect, and they are best evidences for the organic evolution. The third evidence for organic evolution is paleontological evidences. The word paleontology is defined as the study of fossils. The father of paleontology is Leonardo da Vinci. The father of modern paleontology is Cuvier. Fossils or remains or impressions of organisms preserved in the rocks for centuries. And the fossils can be bones or teeth, impressions, casts, or an entire organism. From the study of the fossils, following facts have emerged. There are a large number of species that lived in the past and are extinct now. The extinct species show similarities with the present day forms. For example, the woolly mammoths, whose fossils have been found in the Siberian region, existed about 10,000 years ago. They were definitely elephant, though they may differ from the present day forms. Fossil records show the appearance of new species at different times, making it clear that all organisms were not created at once. Oldest vertebrates are fish and youngest vertebrates are mammals. The fossil evidences reveal a gradual development of organisms with time. Importance of paleontology. Paleontology helps to reconstruct the phylogeny of horse, is the best example of an almost complete fossil record has been found in North American sedimentary rocks. So this we can see in the next slides, the phylogeny of horse or evolution of horse. The chart we can observe in later slides. So the complete evolution of horse, that record is available. The study is completed. Paleontology helped to study the forms and structure of the extinct animals. The well-preserved fossils are also helped to study the habits and behavior of extinct species. Here are some of the examples of the fossils available for the evidences of the extinct species. So here it is the a fish fossil. It is about 45 million years old, and it is another impression of fishes in the rock, fossilized in the rock for thousands of years ago. And it is one more, it is a turtle, that means tortoise, a skeleton of extinct marine turtle which has been put together from it is the paleontologists have determined its size, muscle structure and movement. So and these fossils, the preserved fossils, are helpful to study the habits and behavior of extinct species, how they behave and what are their food habits. And also it is helpful for the study of structural by comparing the present day forms with the extinct forms. The presence of intermediate forms in fossil records indicates the connecting link between two groups of organisms. Example is Archaeopteryx shows the features of both reptiles and birds. This is missed in previous part when we are discussing about connecting links. So the connecting link between reptile and mammal we are given, but we are not mentioning in that connecting links. This Archaeopteryx is the connecting link between the reptiles and birds. So these intermediate forms, nothing but connecting links in the fossil records, indicating that the characters of that. So that means this Archaeopteryx, it is having wings and tail, but the beak concedes the teeth. So presence of teeth is the reptilian character, and bird character having the wings. So that indicates that the birds are evoluted from the reptiles. And this Archaeopteryx is considered as the missing link between the birds and reptiles. It is no more. It is extincted species on the earth. Types of fossils. There are different types of fossils. That number one, intact, not damaged. The pollen is preserved intact because no decomposition occurred due to presence of sporopollen in axine, outer layer of pollen grain. So fossils of microscopic structures are called as the microfossil or maybe palinofossils. And second type, compression. The internal structure of organism is lost and a thin carbon film giving the outline of the organism is left. They are common type of plant fossils. Third type, cast or molds. The material surrounding the buried organism hardens. The skeleton decays, leaving the impression in the form of exact replica of the organism called cast or maybe mold. So it leaves the impression, the mark, the exact replica of that organism. So remaining skeleton and flesh, everything will be decayed and only impression of that organism is left. It is exactly similar to the organism that we will call as the cast or maybe mold. So we may observe some of the molds of the gods. We will get bronze mold, or maybe it may with we will get the mold of idols. So like the impression of that organism, buried organism is left. It is exactly replica or maybe similar to the organism. Fourth type, coprolites. The fossils of fecal matter or rectal content are called as the coprolites. They indicate the type of food consumed by the extinct organism. So fecal matter is nothing but undigested, consists undigested food material eliminated by the organism. So by analyzing the composition of that fecal matter, they find the composition of food what they consumed when they are existed on the earth. And fifth type, petrification or permineralized, slow replacement of the parts of dead organism by inorganic materials like iron pyrite, silica and calcium carbonate. Thin sections of fossils under microscope reveal fine original structure. Sixth type, unaltered fossils. Fossils can be in the form of complete organism, and how we will get that complete organism as a fossil, because they frozen under snow at very low temperature. In such conditions we will get the complete organism fossil. And buried in tar, oil or volcanic ash, in such conditions we will get the complete organism unaltered. There is no any alteration of that fossil. And seventh type we will call it as pseudo fossils. These are not the fossils but give a similar impression as a fossil. At times, mineral substances in rock crevices get crystallized and develop patterns that resemble the flower or leafy outline of a plant. Dating of fossils. Nothing but how can we identify the age of the fossil? That means how many years back it is existed on the earth. To find that, we will call it as the dating of fossils. There are three main methods for the dating of fossils, that is radioactive method, thermoluminescent method, and electron spin resonance method. Radioactive methods. There are again three types. That first one, radioactive uranium, uranium 238. The half-life period during which half of the material is decayed. The half-life period of this uranium is 4.5 billion or 4.5 into 10 to the power of 9 years. After emitting alpha and beta particles, it is finally changed into the stable lead plumbum 206. It can be used for dating the fossil or rock of more than 50 million years old. And the second method, radioactive method, that is potassium argon method, K40. The half-life period of this radioactive isotope is 1.3 billion years. This method has been used for dating the hominoid fossils of Africa. And third type of radioactive method, that is the radioactive carbon method, C14. The half-life period of this carbon is 5750 years. After emitting beta particles, it changes into nitrogen 14. This method can be used for dating recent fossils up to 10 into 10 to the power of 4 years. Second method of dating of fossils, that is a thermoluminescent method. In this method, the rock mineral is heated above 500 degrees centigrade and the trapped electrons are released, creating a flash called thermoluminescence. The intensity of the luminescence can be measured to determine the amount of time when this mineral was last heated. This method is suitable for the samples that range in age from few hundred years to 5 lakh years. This method is suitable for the minerals of quartz, zircon, calcite, etc., which have been previously heated to above 500 degrees centigrade. Such samples include remains of ceramics, bricks, fire pits, kilns, smelters, smelter walls, etc. Third type of dating of fossils, electron spin resonance method. It is also based on the accumulation of trapped electrons in minerals. More the trapped electrons in the minerals, the older the sample or fossil is. It can be used for time periods up to 2 million years. It works best on carbonate minerals. Evolution of horse or phylogeny of horse. The evolution of horses, as evidenced by the fossils, reveals several important trends. The following list identifies the major evolutionary trends of horses. General increase in size, progressive loss of toes, lengthening of toes that are retained, lengthening of limbs in general, enlargement of brain, increase in height, increase in the complexity of molar teeth, and an enlargement of the last two, eventually the last three premolars, until they came to resemble molars. All these are the changes which occurred during evolution of a horse. Here it is the flow chart, a diagrammatic representation of that. So the first in the evolution, the earlier, that is in the Eocene epoch period, 54 million years ago. In that time, the horse height is 0.4 meters. It is about size of a dog. When it comes to the Oligocene period, that is 38 million years ago, that become 0.6 meters. And when it comes to the Miocene period, that is 26 million years ago, the height become 1 meter. And when it comes to the another period, that is a Pleiocene period, that is 7 million years ago, it is 1 meter and little more, means more than, greater than 1 meter and less than 1.5 meter. And when it comes to the 1 million year, that means Pleistocene at present, Pleistocene period, now it is going on Pleistocene, and at present the Rs, it is up to 1.6 meters in the concern of height. Now if you are compare the digits present in the forelimbs, so you can find there are 4 digits in forelimbs and 3 in the hind limbs. Whereas it comes to the second Oligocene period, that is 38 million years ago, it consists only 3 digits. And then come to the next stage, that is in Miocene, 26 million years ago, the digits 2 and 4 reduced and they ran on only digit 3 only. Next, when it comes to the Pliocene period, that is 7 million years ago, the digits 2 and 4 are highly reduced. So this is 2 and 4. That 2 and 4 is highly reduced here. That means completely depends on the third digit. Now when it comes to present day Pleistocene period, the digits 2 and 4 is highly reduced and elongated and strengthened of metacarpals. It gives the strength to the metacarpals. So when you are observed the phylogeny of this horse, or maybe evolution of horse, already we mentioned the points. It is the trends in the height as well as in the digits of the fore and hind limbs, and also their brain capacities and size, body size is gradually increases, and the height is gradually increases, and brain size is gradually increases, and digits are gradually reduces. All these we can observe the complete phylogeny in the evolution of horse. Thank you, students. We will discuss the remaining evidences. There are biogeographical evidences and molecular evidences in the next part.